Good afternoon, everybody. My name is Andrew Alamban, and I work in Dr. Bird's lab. Thank you for being here. And today I'm going to talk about my project is investigating the role of connexins in cell proliferation. So let's get started. So just with a raise of hands, does everybody know what these icons represent? Yes? Cool. So as you all know, these services are very important in our daily lives. They're so important in helping us organize, interact, and communicate with our peers, our friends, just to make sure that we can be on the same page. We have very complex schedules, a lot of things that we want to do, and these help us keep ourselves grounded. So imagine if these services got disrupted or didn't even exist. That would kind of be a nightmare in today's technological society about how connected everybody is. I would like to bring that nightmarish idea into the context of the biological model. So what I have up here is the pathway for the vascular endothelial growth factor receptor, VEGFR. And I don't expect you guys to understand. I don't understand it, not even close. I pulled it off of the internet this morning. But the main point that I'm trying to make is that studying the pathway is really freaking complicated in the context of cell proliferation. And keep in mind that this pathway is just something that's ongoing in one singular cell. So imagine if you're running around and you're a skateboarder or something and you trip and you scrape yourself. And let's zoom in on that wound cut, and suddenly your blood vessels are like, oh my goodness, what are we going to do? There's like stuff coming in, we need to fix the problem. How do these cells know how to coordinate with each other such that they can all agree that, hey, we need to proliferate, or okay, we're good, we need to stop and calm down? So in order to do that, this is where connexins are going to come into the picture, but before I get started with that, I'd like to do this analogy of a cell phone. So a cell phone, as we all know, has its little inside parts. And once you put those parts together, then you have one phone. But what use is a phone if the receiver of your calls doesn't have the cell phones themselves? It's kind of just no. Well, I guess you could look up memes and go on the internet and all that stuff. But that's the point. The point is you need two of them to tango. So similarly, the proteins that we study are connexins, and these are the subunits that will essentially form these structures that we call hemichannels. So those four that's the main protein, and then each barrel right here represents one of these four right here. And then they form this hexameric structure, and once they connect with another cell, then they form this conformation, the gap junction channel, which directly connects one cell to another and allows the exchange of solid materials, metabolites, ions, without ever going outside of the cell, allowing your cells to communicate rapid and quick. Each conformation also assumes its own function. So in the subunit protein level, it could assume this intracellular communication with other proteins within the cell. When it comes to the hemichannel conformation, it can swap out materials inside of itself into its environment, like I believe ATP is a common one that gets spit out. And then last but not the least is the intercellular communication that is so important in what makes your heart beat so rhythmically. I've talked a lot about connexins and the various roles it does. What is its role in proliferation? So what we did, at least for my first year project, was to investigate the role of connexin proteins in connexin deficient cells, which are rat and scleronoma cells. And in this proliferation curve right here, in solid black you'll see connexin positive cells, and in white and dotted lines you'll see the connexin deficient cells. And I'm pretty confident that there is some kind of effect, which is really nice. So the next steps that we're trying to do is the mechanisms. Again, I've talked about the three different conformations that it can assume. Is this cell growth regulated via the protein-protein level, via the hemichannel level or via the gap junction level. It's poorly understood as of the moment, and that's where we're kind of headed in the future towards my project. So for the bigger implication, I would hope that once we understand the mechanisms as to how these connexins are affecting cellular proliferation, then we can kind of zoom back out into this more systems level view of how it plays a role in something like the VEGF system. And hopefully we can kind of take advantage of this knowledge to improve and come up with therapeutic tools for wound healing and possibly tumor suppression. Thank you.